Okay, next we have Jack Towson, and Jack is here in person, and the title of his presentation, Patterns of Population Structure and Gene Flow in a Fragmented Environment, New Road Infrastructure Impacts Bobcats and Ocelot Dispersal.
Okay. Awesome. Well, thank you for that introduction. My name is Jack Towson of the Caesar Kleberg Wildlife Research Institute with Texas A&M University, Kingsville. And today I'm going to be presenting some preliminary research as part of my master's thesis titled, Why Did the Cat Cross the Road? A Genomic Perspective on Felid Behavior. Roads are critical infrastructures of the economy, but they do raise alarming concerns for the wildlife that they impact, especially in South Texas. Species including the bobcat, a widespread generalist felid species, to the ocelot, a nationally endangered specialist felid species, are prime roadkill candidates and are prone to habitat fragmentation and various anthropogenic, genetic, and environmental detriments that could be exacerbated by roads. However, to reduce such roadkill opportunities, recent partnership by the Feline Research Program of Texas A&M University, Kingsville, and the Texas Department of Transportation has helped identify wildlife hotspots along notable roads in South Texas and have implemented wildlife crossings to help promote landscape and population connectivity. But limited genomic research on felid behavior has been completed to understand the decision-making behind road crossing and avoidance behaviors. Bobcats and ocelots were captured using modified box traps at ranches and refuges along US Highway 77 and Farm to Market 1847. We accumulated a fair amount of blood samples dating all the way back to 1988, and DNA from these blood samples was isolated and extracted, amplified using single nucleotide polymorphisms, imaged, and will later on be sequenced. Additionally, collars were fitted and GPS movement data was tracked for all of our individuals. It is worth noting that because bobcats and ocelots share similar life history traits, we can use bobcats as a preliminary proxy for our genomic research due to the limited amount of information surrounding the small, fragmented, and vulnerable population of ocelots in the United States. To incorporate such a targeted gene approach into our research, I completed a functional gene ontology of the bobcat genome and identified 20 genes believed to be involved in some sort of road crossing decision-making. Our most promising gene, MAOA, is a monoamine oxidase gene that regulates aggression and boldness, both of which I believe likely contribute to whether a bobcat decides to cross a road or not. I aligned this gene for 27 carnivore species, including 10 felids, to examine sequence variation with hopes of identifying point mutations that may code for variable phenotypic behaviors. Multiple MAOA studies involving gene knockouts in mice models have identified point mutations in the 8th exon of this gene that evidently cause behavioral shifts in the mouse that do in fact have these knockouts. So we will specifically look into the sequence variation of this particular region across our individuals to see if we also see such mutations. Later on, we plan to assign genomic IDs to all of our blood samples and construct pedigree relationships between our individuals to gain a better understanding of the relatedness across the cat populations of South Texas. Assigning genomic IDs to and constructing pedigrees for our cats is critical because let's say, for example, that we have a roadkill individual who hasn't been collared. Our pedigree analysis could tell us the genomic IDs of this roadkill's parents, and if we collared one or both of these parents, we can then use the GPS movement data for these to identify the parental home range that they inhabited, which would also serve as the origin where this roadkill came from. From there, we can then examine factors that have influenced the spatial route that they took prior to being hit by a vehicle, such as terrain type and overlapping home ranges they may have encountered with conspecifics. Additionally, kinship can also help identify individuals who may be at a higher risk of becoming a road mortality, specifically siblings or offspring of an individual that we know had already become a roadkill. Because of the high likelihood of MAOA's involvement in felid decision-making, we also want to better understand the physiological expression of aggression in felids. One attribute that is recorded for each bobcat and ocelot that we capture is activity or demeanor, and it is measured across a 5-step gradient ranging from calm to aggressive. By quantifying aggression for individual cats in the population, we can then examine the interconnectivity between each individual cat's MAOA genotype, its observed phenotypic aggressive behavior in the field, and its GPS movement patterns to shed light upon the role that aggression plays in social and spatial dynamics, with particular bias towards roads. To initially do so, I estimated evolutionary distances for the MAOA gene across the carnivore species I included in my alignment. Each of these numbers represents the amount of variation of base pairs in the gene sequence between any given two species, and thus calibrates the baseline of variability in the stream for these carnivores. As expected, the closer the two species were evolutionarily to each other, the more similar their MAOA sequences were, as you can tell with the nearly identical MAOA sequences for the snow leopard and clouded leopard. In the same way that we compared gene sequences of different species, we will compare individual Bobcat sequences and individual Ocelot sequences within the population to identify and analyze such points of variation that contribute to these differences in road decision behavior. This individual sequence variation for MAOA is reflected in some way as variation in risk assessment observed in our GPS movement data when a cat encounters a road. In other words, some cats might be more genetically inclined to take a risk and cross a road as compared to some other cats. Having certain point mutations in the MAOA gene, as well as specific point mutations in the other road-related genes, likely contribute to these different barrier behaviors that we have observed in our GPS data. By examining functional variation in felid road behavior genes, their movement patterns will be examined with a new component to help us understand the underlying genomic aspect that drives their decision-making and ultimately contributes to their chance of survival. I plan to further analyze the genetic makeup of the MAOA gene, as well as the other road-related genes that we have yet to look into, and their influence on felid movement patterns, taking into consideration environmental factors such as social tolerance, habitat terrain, home range size, and fecundity, but also anthropogenic factors such as vehicle speed, highway noise, and traffic volume, all of which could potentially contribute to road crossing probability. By homogenizing functional genomics with our movement ecology, informed decision-making regarding the wildlife crossings in South Texas can be maximized to implement the survival of these beautiful felid species, despite the challenges that roads have and will continue to present for their survival. With that being said, I'd like to take a moment to thank the Texas Department of Transportation for funding all the research in this talk, as well as the members of our lab that have helped with data collection and initial analyses. If you're interested in learning more about our research, please feel free to check out our page, the Feline Research Program page on the Cesar Claybert Wildlife Research Institute website. And with that being said, I'm hoping we have a minute or two for questions, and I'd be happy to answer those.
Thank you for your presentation. I'm Bobcat. So doing this, I feel like I'm home. So I want to find out, you guys will compare Ocelot, Bobcat to a mountain lion and see how the aggression and differences will be.
Yeah, so the question for the people online is, are we going to be comparing Bobcat and Ocelot sequences to mountain lions that also inhabit South Texas? As far as I know, currently we're just going to focus on those two species. We could definitely look at the sequence variation in the alignment that I was talking about earlier. The mountain lion was included as one of those felid species, so we could look at some variation in the MAOA gene that we've already looked into, as well as those other 19 genes that we think could be worth looking into to try and understand what's going on with their road behavior. Yes, sir.
We have a lot of time, we have one more presenter, but just to keep it equal, I'm going to just move on, and then Jack will go around.
Yeah, I'll be around if you have any more questions.
There'll be plenty of time to